So, welcome back, everybody, to the second session of this extremely interesting meeting. And, as you know, this is the second international symposium of the Society for the Study of Evolution, organized in collaboration with the Indian Society of Evolutionary Biologists, ISEB. In this session, we will have a last talk, and then we'll have an open discussion about the challenges and opportunities of conducting research in evolutionary biology in South Asia. So my name is Sutirth. I am a member of the editorial board of Evolution journal. I'm also a member of the executive committee of the Indian Society of Evolutionary Biologists. So now I would like to welcome our next speaker, Riddhiman. Riddhiman Dhar is an assistant professor at the Indian Institute of Technology, Kharagpur. He studies the influence of genetic diversity and non-genetic heterogeneity on genotype-phenotype mapping. Riddhiman will speak today on how neutral mutations impact adaptation to new environments. Riddhiman, over to you.
Thank you, Sutirth. I'd like to thank the organizers for this opportunity to present our work here. I'll start with sharing my screen and the presentation. So hopefully the presentation is visible.
Yes, Riddhiman.
Okay, great. Okay, so today I'm going to talk about how neutral mutations impact adaptation to new environments. So I will show that these neutral mutations have very complex roles in case of organismal evolution. So neutral mutations have been studied for quite a long time, and they have been proposed to facilitate evolutionary adaptation to new environments. What we did in this work is that we asked whether neutral mutations do the same in case of organisms, complex organisms. So I'll start with acknowledging the people. So this is our lab, and the work that I'll be presenting today is the work done by the graduate student Shreya Raut in the lab. And this work is also done in collaboration with Professor Riddhiman Dhar at the University of Exeter, and our group, Dr. Richard Lindsay, who's a postdoc in Kharagpur. I'd also thank the funding agencies at Kharagpur, CRB India, and also Newton Hub PhD Placement Part, which actually supported Shreya to go to the UK and do some of this work there. So I'll start with the neutral mutations. So what are neutral mutations? So neutral mutations are those that do not have any impact on fitness. Now, as we talk about fitness, it means there is some specificity about environment, right? So these fitness measurements, they are done on specific environment, and in that environment, these mutations do not show any effect. Now, if we change this environment, these neutral mutations might show some fitness effects, and which means there is some sort of hidden evolutionary potential, right? So when the environment changes, these mutations may start showing some fitness, and this might help in adapting to some new environmental conditions. So that's why these mutations are also referred to as cryptic genetic variations. So in one environment, they are not seen, right? They do not have any impact on the phenotype, but when the environment changes, they manifest their phenotypes. So I'll illustrate this with this genotype network, where each node here in this graph is a genotype, and the nodes that are connected to a genotype can be reached by a single mutation. Now, it has been proposed that neutral mutations can facilitate evolutionary innovation. So you can think about this in simple genotype network terms. So we start with genotype here, and we can have neutral mutations. So we are traversing through this genotype network, but the phenotype does not change, right? So the environment remains same, the phenotype does not change. So the genotype or the organism is able to explore a lot of genetic diversity without affecting the phenotype. Now, at some point, there might be some genotypes that actually can give rise to new phenotypes, and these might be helpful when there is an environmental change, right? So this red phenotype or the green phenotype, they could help environments, could help the organism to adapt to these new environmental conditions. So this idea has been tested in ribozymes and proteins. So I'll show the results from this work, which was done on a ribozyme by Hayden et al. In 2011. So what they did is they evolved this ribozyme population in their native environment, which means they were selecting for native function of that ribozyme. So they performed several rounds of this neutral evolution where the ribozyme molecules were accumulating mutations, but there was no change in function. And subsequently, they selected these ribozymes to new environmental conditions. So they imposed selection for new function. And what they observed was very interesting, right? So the native ribozyme that did not go through the neutral evolution was actually adapting much slower than the ribozyme molecules that actually experience neutral evolution. So they also explored the genotypic basis, right? What are the mutations that actually helped in adaptation in these neutrally evolved ribozymes? Similar idea has been tested in proteins also. So there are examples of two proteins where this has also been seen. So the first work actually was done on YFP, and the second one was done, the second work, cryptic generation in enzymes, that actually was done on metal-beta lactamases. So what the authors showed is that different classes of metal-beta lactamases, these are orthologous proteins, and they actually showed different evolutionary potential because of the genetic variants that they harbored. So the question that we started with is that, okay, we see this principle hold in case of ribozymes as well as for proteins, but does the same principle hold at the organismal level, right? So what happens if we do the same experiment with an organism, right, instead of a single protein or a single gene? So the question then becomes, right, does neutral evolution facilitate adaptation to new environments at the organismal level? So to test this, we actually did laboratory evolution experiment in Saccharomyces cerevisiae. And here is the scheme for the evolution experiment. So we started with the ancestor strain, right? And we divided this ancestor strain into three subpopulations, A1 into A3. And we let them grow in rich media for 10 generations. So they had all the nutrients in abundant supply, and they were growing happily. So after 10 generations, they were then transferred to fresh media. And this process was repeated for 1,000 generations and will denote these lines by the N1, N2, N3 lines. So they undergone this neutral evolution. So we represent them as N1, N2, N3, and we'll denote the ancestral lines as A1, A2, A3, and I'll show the results for these lines. Now, the first question that we asked is whether there is any change in fitness in native environment over these 1,000 generations. Okay, so this is very important. So we followed two processes to measure that. So we first performed the computation assay between these A and N lines. We take strain, which gives us red color, so we can compute both these lines and kind of measure, right, what fraction of cells come from A or come from this red strain. And in addition, we also did the growth curve analysis of these A and N lines, because computation assay can capture the fitness only at a certain time point. So we're doing this only for a certain time point, but these evolution experiments can lead to many complex adaptations. So when you do the computation assay, we see very little fitness change in native environment over 1,000 generations. So here you see the relative fitness, y-axis is relative fitness compared to the ancestor, and x-axis is generation. So at 1,000 generations, you can see there is a slight increase, but that's not significantly different from what we started with. And this is true for all three replicates. Now, as we measure this competitive fitness in just one time point, so what we want to do is still want to do this growth curve analysis. So y-axis is the absorbance and x-axis is time. And what we see is very interesting is that there is some sort of adaptation, although not a lot, but we see slightly shorter lag phase in these neutrally evolved lines. But the carrying capacity, the saturation actually is reached at L1 level. This holds for all the three lines in 1, N2, N3. And we estimated these parameters more carefully, right, based on the growth models. And what we observe that growth rate does not change, but we observe a decrease in lifetime, also a decrease in the carrying capacity. Now, the next question is, so in native environment, we do not see much change, but the next question is, what happens if we now expose these end lines to new environments? Okay, so do end lines show higher fitness in new environments? So we did that, right? We exposed these A lines and N lines to different media, different environment conditions. So we subjected to minimal growth media, which contains only essential nutrients. So kind of like limiting the amount of nutrients. We also exposed them to some stress, osmotic stress and oxidative stress. In addition, we also tested against some antifungal compounds. So what we see is that end lines show longer lag and lower growth in minimal growth medium. So which means they kind of probably have accumulated some mutations that allow them to do without certain nutrients, or they might have some mutation that actually lead to loss of function in some of the genes that might be required in survival in this minimal medium. So as you can see, this N1 in 3 lines, they're showing a lower growth rate. On the right, you can see the estimates for growth rates, lower growth rate as well as longer lag time, especially this N2 line, and also they reach lower maximum concentration. And we also measured this for other conditions. I'll show you only some results. In hydrogen peroxide under oxidative stress, we see significant decrease in fitness for these neutrally evolved lines, N1, N2, N3. And as you can see, N2 line actually stopped growing. It was not growing at all in this concentration of hydrogen peroxide. And in addition, what you will see for this N1 and N3, we see a lot of heterogeneity in the measurements, right? Because we're doing this in multiple technical replicas and they were showing a lot of variation in that growth rate. Nevertheless, what we see is that lag time has increased substantially for N lines, and also the carrying capacity is lower. But interestingly, what we see is that N lines exhibit longer lag but higher growth rate in antifungal compounds. So this was true for both antifungal compounds. I'm just showing the results for one antifungal compound, caspofungin. So what you see is that they kind of N lines tend to have a really long lag phase, but then once they start growing, they grow really fast and they reach actually higher concentration. Right, higher saturating concentration. So you can see that growth rate is actually higher, but the long lag time is longer. And these are actually very long time scales. As you can probably see, it's around 60 hours. This is what is happening. Now, we have seen, right, this is kind of like a mixed results. Right? There is no clear pattern that inlines are better or worse. We see that the fitness in new environments depends on the environment, right? So we measure in different environments. In some environments, inlines are worse than A-lines, whereas in some environments, inlines are better. Now, the next question is, do inlines show better adaptation to new environments, right? So it's not always necessary that this initial fitness will dictate the adaptation with these new environments, right? So initial fitness may or may not predict the degree of adaptation. So to test that, what we did now is we took those A lines and N lines, and then we exposed them to these stress environments. Okay? So the same stress environments that we used earlier. And what we did is like we let these cells grow for 48 hours and then transfer them to the fresh medium, again containing the stress, right? So we did this for all 6 lines and for about 10 rounds. Now, what we see, the question is, do N lines show better adaptation, right? So when we measure this growth in the stress environments during the evolution experiment, we did not see any better adaptation in N lines. And what we observed that N lines sometimes also pass through lower fitness paths. Okay, so what you see here is the fold growth along the y-axis. So it's kind of just a amount of growth in certain 48 hours, and the x-axis is the rounds of evolution, and the blue line is for the A lines and N is for the N lines. So what you notice is that there is almost no difference in the adaptation to these stressors. And in these two antifungal compounds, what you'll see is that end lines actually pass through lower fitness values, right? So here are the fitness values of end lines versus rounds, and the end lines actually pass through lower fitness values than the A lines. Very interestingly, in hydrogen peroxide, it was like varying really widely, and what we observed is that two replicate populations of end lines actually became extinct after certain rounds. So N2 was not growing at all from the beginning, right? It went extinct right at the start. And a few rounds later, the N1 line also went extinct. Okay. However, N3 line actually persisted for 10 rounds. Now, the question is, what is actually driving this, right? So to summarize what we have seen so far, so the question that we started with is, does evolution facilitate adaptation to new environments at the organism level? So we have seen it's not necessarily true. And in some cases, it can even lead to extinction. Okay, so as we have seen in the case of hydrogen peroxide. The next question we had is that what role do mutations play in determining the outcome, right? So we see this kind of adaptation differences between A lines and N lines, and also some extinction events. So can we find out the mutations that are probably responsible for this process? So to do that, we actually sequenced these A lines and N lines and identified these single nuclear polymorphisms and indels. And we have not done the complementarity analysis yet, so I will not be able to show that data. But I'll show you the single nuclear polymorphism and indel data. So what we see is the replicate N lines show distinct mutational patterns. Okay, so in this circle, you can notice the number for the chromosomes, and each track shows the mutations that are present in these three lines. So the first tag is for N1 line, the second is for N2, and the third is for N3. So as you can see, most of these mutations are unique, except some shared mutations here and there between the lines. What you will probably notice here is that one of these lines, N2 especially, actually contains a very high frequency mutation in the mitochondria, and I'll talk about that a bit later. I have also plotted some of the mutations that are present in the ancestral line, but were subsequently removed from this N line. So, for example, this mutation, this is shown in gray. This is present in the ancestral line, but was absent in all the default lines. So if we look at the overlap between these mutations, there are only few mutations that are common across these lines. So we see about 7.4% overlap in terms of mutations that show population frequency greater than 5% between these N1 and N2. For N1 and N3, we see about 4.5% mutations. And I've also highlighted some of the genes that we know are involved in stress response or mitochondrial function among the lists. In N2 line, very interestingly, remember this is the line that went extinct at the beginning of this experiment in hydrogen peroxide. It harbored a very high frequency mutation in the gene cytochrome B. This is a mitochondrial gene. And since mitochondria is involved in adaptation process, right, in oxidative stress and other stresses also, this could be one of the contributing factors for extinction event of N2 lines. If we plot the number of mutations and also look at the distribution of synonymous and non-synonymous mutations, this is what we see, right? Non-synonymous mutations are much higher than synonymous mutations. There are also some promoter mutations that we could map. And when we calculate the DNTS ratio, we see values much lower than 1. And here some mutations are just highlighted, which we think would be interesting. So for example, as in SLM1 gene, there's a non-synonymous mutation at a frequency of about 35% in the population. This gene is involved in stress response in yeast. For N2 line, I have already mentioned, this is the cytochrome B gene, about 80% frequency. This mutation is present in the population. And for N3 line, this HKR gene, this mutation is present at 76% frequency. And this is a gene that's also involved in stress response. This gene acts as an osmosis agent. So to summarize the results, so at the organismal level, what we have seen, mutual evolution can make populations less fit in new environmental conditions. And but we have also seen that mutual evolution does not always lead to better adaptations in environments, and in some cases can even lead to population extinction. And replicate populations take independent evolutionary paths that is dependent on the mutations that are arising in the population. Here are several open questions still, right? So the first question is, of course, why do the lines in one and N2 become extinct under oxidative stress? So we think that this is because of, in N2 line at least, because of the mitochondrial mutation, and in N1 line, maybe there are some mutations that might be involved. So for example, in N1 line, we see a mutation in a gene that's involved in DNA damage response, and that would be a possible reason behind extinction events. But then this is speculation, we have not tested that yet. What we have tested is not just due to reduction in mitochondrial activity. So when you measure the mitochondrial activity of the N lines, we do not see any change in the mitochondrial activity. In fact, they are slightly better in the ancestral lines. The second question is actually more general and probably more interesting, is why are the outcomes different at the organismal level compared to a ribozyme or a protein, right? So this is something that we still don't understand fully, right? Maybe there is, because this, or at organismal level, the whole process is much more complex, right? So we have multiple genes, their interactions, etc. This could also be because of mutation, right? So if you look at the earlier studies, some of these mutations were introduced by PCRs, right? So these are kind of artificially introduced, and the mutation rates are much higher than the natural mutation rates. Whereas in our experiment, we are using natural mutation rate of yeast. We are not giving any mutagen or kind of changing any polymerase to induce higher mutation rate. In addition, the target size is much larger, right? So we have the whole genome for the mutations to take place, whereas those earlier studies, they were focusing on one gene, and the target is just one gene or one ribosome only. So we are still we still need to explore this a bit more, but here these are a few directions which we can take. So to summarize, at the organismal level, we have seen that the impact of neutral evolution on fitness and adaptability is much more complex compared to a single ribozyme or a protein. And the phenotypic outcome is dependent on the specific mutation events and the environment that we set, right? So the new environment that we impose also determines the phenotypic outcome. Thank you.
Thank you, Riddhiman, for that extremely interesting talk. It was great, and also we have quite a bit of time for the question and answer session.
Okay.
So Deepa has asked something. She says, I'm not sure that your initial evolution was neutral. Looked like there was about 10% increase in fitness and change in lag, etc. Can you comment?
Yeah, so we see about 5% change, right, when you do the competition assay. And also there is change in lag, right? But again, remember, so when we're doing the competition assay, we are measuring this outcome only at one time point, right? So especially when in our assay, we are doing it around late log, right? When you're taking the cells and plating them and measuring their fitness. But if you look at, so again, this fitness would depend on when we are sampling, right? So if we sample at stationary phase, that will actually tell us, that will show that, okay, these ancestral lines are actually fitter than the neutral lines. Okay, so that is something I think we probably have to think about more deeply, right? How actually, what would be the exact fitness that we talked about, right? Whether it's just growth rate, whether it's also the carrying capacity, also the lag time, right? So the competition assay gives us this fitness value only at one time point. The other point is that we have also looked at mutations, right? So we see like when we calculate this DLTS or the K-case ratio, we see values much lower than 1. So what we think is mostly purifying selection that is going.
Okay. So Shamana Kar is asking, why do the neutral mutations even accumulate if they are often detrimental to the organismal level?
Yeah, so that's a very interesting question, right? So, as the term suggests, so neutral means these mutations do not have any fitness effect in the rich growth medium, right? So, in the evolution medium, rich growth medium, we do not see any fitness effect, right? These mutations have no fitness effect, so that's why they accumulate, right? They are not detrimental in that environment. Only when we change the environment, we see these fitness effects, and some of them turn out to be detrimental.
Pratibha Sanjanbam has something to ask. She's saying very
Mhm.
interesting. However, I find very surprising that N lines have higher growth rate compared to ancestor in antifungal compound. But when these lines are exposed to new environment, they behave similarly with ancestor. Could you please clarify if I missed anything?
Yeah, so so there are two parts, right? So when you do the initial fitness measurements in the new environments, so there we see higher growth rate but longer lag period, right? So within that short period of time, what we think is probably there is some sort of physiological adaptation is also important, right? So that's why I also mentioned the time point. We were doing this experiment for, let's say, 100 hours, right? So around 60 hours. After 60 hours, we see this growth in the N lines, right? So which means there is also some sort of physiological adaptation that is happening in those lines. And the second part is the evolution or the selection experiment, right? So where we adapt these N lines to antifungal compounds, and only then we see this difference in fitness. So they are not behaving same as ancestral line. There is some difference, and they're actually going through some low fitness parts. I hope that answers your question. There are two parts, right? One is the initial fitness measurement. The second part is the selection experiment.
Okay. Do we have any more questions? Pratibha says that does answer her question.
Okay.
Okay. So if nothing else is coming up, can I ask you something, Riddhiman?
Sure.
So the number of rounds of mutations that were done or transfers that were done were about 10, I think, right? 10 rounds
Yeah,
of
10 rounds in
evolution.
selection. Yes.
Yeah, 10 rounds of selection. Isn't that a bit on the lower side? Because this is neutral environment, or at least very low pressure selection pressure environment. So obviously things are happening really, really slowly. So if you expect mutations to kind of settle down and rise to some kind of a frequency, you need a little bit of a longer time span for the evolution. What is your view on that?
Okay. So actually, we are doing the neutral evolution for 1,000 generations, right? So the first phase where we allow cells to accumulate mutations, right, in rich growth medium, that's about 1,000 generations. And then
Okay.
we do the selection experiment where we then adapt them to new environmental conditions that last for 10 rounds. Okay. So
So 10
if
rounds is how many generations?
so I'm not saying generation because it's very extremely difficult. Because in the stress environment, depending on also the condition, this number of generations are different.
Correct, but roughly speaking, roughly.
So roughly maybe 100 generations. 100 to 100
Okay,
to
so that will be a bit on the lower side, right? Well, given that we are dealing with microbes.
Actually, so the idea behind the second experiment is that what we are trying to see is that whether the mutations that are accumulated in the first phase, whether they are helping in adaptation, right? So we are not, trying to get new mutations in the pool. What we're trying to see is that these mutations are already present in the first phase of evolution, and they should come up, right? If they are present, they will come up and they will help in much faster adaptation.
Okay.
So, yeah, I hope that answers your question, right? So the second
Yeah,
phase,
that is why you have kept it very small. The duration you have kept it small.
yeah,
But as a result of that, you are also getting largish error bars. So I was wondering if it were a little longer, another 100 generations maybe, if the error bars will come down. And if the error bars come down, then things that are not significant will turn significant.
Probably yes, but even then, N lines are lower, right? If you see that N lines are showing lower growth than the A lines, so this is something that we were seeing. And then, of course, for the hydrogen peroxide lines, we are not able to do any experiments, right? Because they are extinct. Two of them are extinct already. And that is also very surprising that even with only 1,000 generations, we have accumulated some mutations that are driving extinction at the end.
Yeah. We can discuss this outside,
Sure,
but
sure.
certainly extremely interesting, bit of work.
Thank you.
Okay, so are there any further questions? We'll wait a few seconds to see. No? Okay, then we have, we'll thank Riddhiman for this extremely interesting talk, and we'll now take a short break of about 15 minutes, I guess, and we will come back at 2:15 for a general discussion on evolution. And yeah, I think that's pretty much all for this particular session. And we'll be back in the next one for a general discussion on evolution in India and how one can connect with the SSC related. Okay.
Okay, thank you.
Thanks. Thanks.